My name is Jovia Katuhaise, and I am going to be your teacher for biology today. As I promised last time, that today we are going to look at the effect of osmosis in plants and animal cells. Why should we study osmosis? The last time I was here, we looked at the major definitions of osmosis, what osmosis is, the major definitions such as hypertonic, isotonic, hypotonic, water potential, osmotic potential, and then we summed it up all together by looking at the significance or the importances of osmosis in nature or in life. But then today we want to go a little bit deeper into osmosis by looking at why should we study osmosis anyway? What is the effect of osmosis? How does it affect our body system? Osmosis can affect the contents of our body if we do not monitor the amount of salt we take in our food or the amount of sugar we take within our tea or the different fluids that we take, or if we have severe conditions such as diabetes mellitus, whereby you have high blood sugar levels. So it has a major effect in our body if it's not monitored, if we do not monitor the intake of salts, intake of water, intake of sugars in our bodies. And that is why we're here today to see what is the effect of osmosis on our red blood cells and what can be the effect of osmosis in plant cells. So today's expectations of the lesson, one, we are going to look at the major definitions. Again, I'm just going to do a simple sum up of these major definitions, such as water potential, osmotic potential, hypertonic, isotonic, hypotonic solution. Then we shall look at crenation, Hymolysis or hemolysis, and then turgidity, flaccidity. Then we shall also have then we shall go into deeper details of effect of osmosis on red blood cells. And then we shall look at effects of osmosis in plant cells. And then lastly, we shall look at the importance of turgidity in plant cells. So those are the expectations of today's lessons. But again, today's reference question is going to be UNEB 2015, question number 31. That is our reference question for today's lesson. It's a graph work. It's a graph work that is taking 20 marks. And it's summarizing what we are studying today. Particularly, it's summarizing osmosis in red blood cells. So if you're able to plot that graph, you're able to describe it, explain it, and attempt the different concepts of osmosis on that number, then you'll have understood what I have taught today and what I taught last week. So to start with, the last time I was here, I said osmosis is a type of diffusion, because when I'm just introducing movement of materials in and out of cells, I said we have key major processes that we look at. One of them I started with was diffusion, telling you that in diffusion you look at movement of molecules or particles from a region of their high concentration to a region of their low concentration. As simple as that. We call it down the concentration gradient. But then when I came back last week, I said there's another form of diffusion we call osmosis. And here we look at the concentration of water or the concentration of solutes in a given solution, and how water can move from one solution to another. Whereas in diffusion, we are looking at particles or molecules moving. In osmosis, I said it's only water that moves, or we use the word solvent. So in your definition of osmosis, last week we said it's the movement of water molecules from a region of their high concentration to a region of their low concentration across a semi-permeable membrane. And I remember telling you that the definition seems easy, but it is always the most difficult definition to get a mark on, because at least students will forget a few things. Like, it's only knowing that we talk about water or solvent molecules alone moving. knowing that they should move from a region where they are highly concentrated to a region where they are of low concentration, and not forgetting the word semi-permeable membrane. And then if I could reminisce a little on what I said last week, I said the osmosis is a little bit different from diffusion, but the concept is the same. The only unique difference here is in osmosis is only water that moves, and in osmosis there must be a semi-permeable membrane that you're not going to find in diffusion. And then I remember saying that osmosis is just part of diffusion. So before I look at the effect of osmosis in red blood cells and plant cells, I just want us to look at a few terms. The first term being water potential. Water potential simply meaning the amount of water within a substance. If you have a lot of water in your body, if a system has a lot of water in it, then it has a high water potential. If a system has less water in it, then it has a low water potential. That takes me back to my tea, the cups of tea that I talked about last week. That if you have 2 cups of tea, and in one you put only 1 spoonful of sugar, and then the other you put 4 teaspoonful of sugar, then the one that has 1 a spoon of sugar is going to have fewer solutes. The sugar solution is very low, and therefore if few water molecules will dissolve the sugar crystals, and that cup of tea is going to be more dilute. It won't be as sweet as the one that where you placed 4 teaspoonful of sugar. So that means that in the cup of tea, so we are having 2 cups of tea. One is A and another one is B, and they are separated by a semi-permeable membrane. Here we put 1 teaspoonful of sugar, there we're putting 4. So here, because the sugar crystals are few, the water concentration is very high. And here, because the sugar crystals are very many, the water concentration is very low. So this one is dilute. This one is concentrated. It has concentrated sugar crystals. So this is more sweeter than A. What does that mean? That the dilute one, we said it's called isotonic, hypotonic. And then the concentrated one is the same as hypertonic. So hypotonic is synonymous to dilute, and it's also synonymous to a substance or a system that has very many water molecules in it. So this one here has a high water potential than B. Why? A has more water than the sugar crystals than B, which has more of the sugar crystals than the water. So the water potential of A is higher than the water potential of B. So therefore, because there's a lot of water in A than in B, osmosis is going to take place by water. Remember we said in osmosis it's water that moves. By water moving from a region where it is highly concentrated across a semi-permeable membrane to a region where the water concentration is very low. So these arrows here are showing the direction of osmosis, moving from A towards B, because in A there is more water molecules compared to B, where we're having more sugar crystals. So I really wanted to have a recap of this, because I am going to use it a lot when I'm talking about the red blood cells and the white blood cells and the effect of osmosis in them. So we ought to know that a dilute solution has fewer solutes in it, and high water content in it, and therefore it has a high water potential, and we call it a hypotonic solution. And then a very concentrated solution has more solutes in it, less water in it, and therefore it has a low water potential, and then it is called a hypertonic solution. And then if we could reminisce on the definition of osmosis, we can define it in terms of water potential, in terms of hypotonic, in terms of water. That osmosis is the movement of water molecules from a region of high water potential to a region of low water potential across a semi-permeable membrane. That's a definition. The next definition could be osmosis is the movement of water molecules from a hypotonic solution to a hypertonic solution across a semi-permeable membrane. That is the second definition. The third definition could be osmosis is the movement of water molecules from a low solute potential to a high solute potential across a semi-permeable membrane. Now that is in terms of solutes. Then the last definition could be is the movement of water molecules from a region of their high concentration to a region of their low concentration across a semi-permeable membrane. More than three, towards there are almost 4 definitions. You just have to understand the concept, and then you don't need to start stammering around in an exam. The moment you get this concept, then you can easily do my number that I am leaving behind today, and you can easily explain the red blood cell, plant cell relations with osmosis. And now these are the key terms that I wanted us to look at. But then there are other key terms before I go to osmosis in red blood cells that we ought to know. The first key term is hemolysis. In some books, you find it written as hemolysis without A. Still, it's okay. Hemolysis coming from 2 words. The first word being hymo and the second word being lysis. Lysis essentially means to burst or to split or to disintegrate. And hymo coming from the word hemoglobin. So this is common in red blood cells, whereby when red blood cells are placed in distilled water, when they are placed in distilled water, I would request the producer to put the other pic on your screens to show hemolysis. When they are placed in distilled water, water will enter into them by osmosis, and then they will extend, and later on they will burst. So when they burst, they will release their hemoglobin into the surrounding water, and once they release the hemoglobin, the surrounding water turns red, the color of red blood cells, and then the red blood cells are said to have undergone hemolysis. So hemolysis is the lysis of red blood cells when placed in very dilute solutions or in hypotonic solutions, causing them to release their contents or hemoglobin into the external environment. That is an example of hemolysis, as you're seeing, whereby you have test tubes. The first part is where you're having test tubes containing blood, whole blood. But then, as you can see, the upper surface is more colorless and the lower surface is red. So that when you get your blood, they can take it through a centrifuging machine, and then the centrifuge will separate plasma from the blood cells, and then when they are separated, the blood cells which are heavier settle at the bottom, and plasma will move at the top, which is kind of colorless. Then the second one, we are having blood cells where we're having the yellow, then it keeps on changing. That one, it's showing that the rate at which blood cells will keep on bursting, changing the color of the solution in the test tubes. So the last test tube is showing red blood cells that have all of them have completely bursted, and the entire test tube is red, showing that there is hemolysis, what I am talking about. I'd request the producer to put on your screen the second thing about red blood cells. That one, we shall get back to this when we come to osmosis in red blood cells. So that is hemolysis, bursting of red blood cells to release hemoglobin. Then the next major term I want us to understand is crenation, because I am going to talk about them more. Crenation now also is in red blood cells. In crenation, the red blood cells, instead, they are not placed in dilute, in hypotonic solution, but rather they are placed in hypertonic solution, very concentrated solutions. And then water will move out of the red blood cells into the concentrated solution by osmosis, and then the red blood cell will reduce in size or shrink. So it is almost the same as shrinkage. The red blood cells will shrink, then they will reduce their biconcave disc shape structure, and when they lose their biconcave disc shape structure, then they become crenated, or they change shape. I would request the producer to put that onto your screens. Now this is what I am talking about. I'm talking about the red blood cells in hypertonic solution, in isotonic solution, and in hypotonic solution. Now the first one is we're having hypertonic solution. A hypertonic solution, it means that the red blood cells are placed in a solution that is highly concentrated, or that has a lot of solute molecules. So when you place a red blood cell in a solution that has a lot of solute molecules, the cytoplasm of the red blood cell contains more water than the surrounding solution, and therefore water is going to move out of the red blood cell by osmosis into the surrounding hypertonic solution, and the red blood cell will shrink or reduce, will start reducing in size due to loss of water by osmosis, and then it will shrink, and that shrinkage will result into it changing its shape from the normal shape you're seeing in the other two pictures, which is biconcave disc, to a more crenated form. So the shape you're seeing that is more like spikes, spike shape, is a shape of a crenated red blood cell in hypertonic solution. So that, and the direction of water at the bottom you're seeing with a green color, that in hypertonic solution in crenated red blood cell, water moves out of the red blood cell into the external environment. Then we are having the middle one where we're having isotonic. Remember we said in isotonic solution, the concentration of water and solutes is the same in two systems. So here, the concentration of water and salts in the red blood cell, inside the cytoplasm of the red blood cell, is the same as that outside the red blood cell. And so there is no net movement of water molecules from one region to another. That means that there is no osmosis taking place in isotonic solution. That is why you're seeing that that middle cell, the arrow we have in green, water coming out, water entering. There is no net loss of water by any of the systems. The red blood cell does not lose water, neither does it gain from another system. So that is how our blood is supposed to be, that our blood is kept intact by having the our blood, internal blood environment, the surrounding plasma having the same concentration as the red blood cell, to prevent crenation and hemolysis. Then we are having the third one where we're having hypotonic. Hypotonic meaning that the red blood cells are placed in a system that has more water than them. So you can see there is one where we're having some blue spills coming out of the red blood cell. So hypotonic, the red blood cells are being placed in a dilute solution. And because the dilute solution there now is having more water, and the concentration of the cytoplasm of the red blood cell is very high, meaning the red blood cell is having more solutes than the surrounding water environment. As such, water is going to move from the hypotonic solution, from the external environment, and then it is going to enter into the red blood cell, as you're seeing using the yellow arrows showing water entering into the red blood cell. So water is going to move from the external environment, it will enter into the red blood cell. The red blood cell will keep on swelling, and later on, when the water is too much, it will burst, and that is what you call hemolysis in hypotonic solution. So that is what I want to talk about, hemolysis and crenation. And then the other major term is in plant cells, we have our turgidity, tugger pressure, and then you also have wall pressure. Turgidity in plant cells. I would request the producer to put it on your screens, showing plant cells in hypertonic, isotonic, and hypotonic. Yeah. So when you look at a plant cell, when it's placed in a hypertonic solution, a solution that has a lot of water, a lot of solutes in it, or a more concentrated solution, water is going to move out of the plant cell into the surrounding hypertonic solution, and as such, the plant cell is going to change its shape. It will become more flabby and soft because of loss of water, and then that plant cell is said to be plasmolysed. Then the extreme end, we are having a turgid plant cell placed in a hypotonic solution. If a plant cell is placed in a solution that has more water than the inside of the plant cell, then osmosis is going to take place, whereby water will move outside of the plant cell from the hypotonic environment into the plant cell by osmosis, and the plant cell will keep on increasing in size. Then it will become hard and stiff, but it will not burst because of the rigid cell wall. So because of that, the plant cell becomes turgid, and because of that, it is stiff and hard. It can make the plant to stand erect onto the ground without even having wood in it. The middle section is where we are having a plant cell placed in isotonic solution, the solution whose water concentration and solute concentration is the same as that of the plant cell, and thus there is no net movement of water. As you can see, it's neither plasmolysed nor turgid, but then it is flaccid in such a way that there is no tightness, that much pushing and tightness of the plant cell because of much water entering it, and then there is not that excessive loss of water. So there is no net movement of water in and out of the plant cell due to the internal plant cell environment having the same concentration of water and solutes to the external plant environment, which is the ideal situation every living cell should be living in. So tugger pressure, turgidity, I've talked about it. Tugger pressure is when water enters the plant cell by osmosis. Like in the last section of hypotonic solution, water will enter, and then later on the cytoplasm, together with the contents of the cytoplasm, the nucleus forming the protoplasm, will start pushing against the wall of the cell, causing to make it to expand to accommodate more the incoming water. So that pressure by which the inner contents of the plant cell uses to push against the cell wall of the plant to extend to accommodate the incoming water is what we call tugger pressure. It is always very high when a plant cell is placed in hypotonic or in dilute solution. And then wall pressure is an equal but opposite force to tugger pressure. So when the water is pushing, the cytoplasm of the plant cell is pushing the cell wall to extend to accommodate the incoming water, the cell wall of the plant is more rigid. It will react by sending an equal but opposite pressure, pushing also the cytoplasm and its contents back. So when this wall pressure and the tugger pressure are equal, the plant cell becomes turgid. So that is what I wanted to talk about, the key terms: hemolysis, crenation, turgidity, tugger pressure, and wall pressure. And now let us look at osmosis in red blood cells. Red blood cells are very important in our body because basically they are found in the bodies of animals. And we all know that they have a biconcave disc shape. And then we also know that they contain hemoglobin pigment, an iron pigment called hemoglobin that has a high affinity for oxygen. Therefore, the red blood cells are very important during the transportation of oxygen within our body through blood. So when you breathe in oxygen, and it diffuses across your respiratory surface in the lungs. It will enter into the bloodstream where it will bind to the hemoglobin pigment in the red blood cell, and then it will be transported by the red blood cell, by the hemoglobin in the red blood cell, through to the different body parts that need it. So therefore, red blood cells are necessary in the transportation of oxygen in our body. But then I want you to know that the blood is divided into components, two components, as I said earlier on. I request the producer to put that picture on your screens. We have the liquid part of blood we call plasma. And then we also have the blood cells. The initial one, not that one, the other one. So when they collect blood from, that one should be placed on the screens. When they collect blood from you, as you're seeing in a test tube, the first illustration where we're having red, very red test tubes, they collect whole blood from you, or maybe by Red Cross people. When they collect whole blood from you, they are going to take it, they will take it to the laboratory, and by use of a machine we call a centrifuging machine, the blood is spinned to separate its contents. When it is spinned, the plasma separates from the blood cells. As you're seeing in the upper test tubes, as I said earlier on, we have the upper colorless liquid part, as you see where the blood is stopping. The upper colorless liquid part and the bottom part that is red. So the upper part is the liquid part of blood, it is plasma, and then the bottom part we're having more of the blood cells. And because it is red, we have more of the red blood cells there. So in your blood, as blood is circulating, the liquid part of the blood and the blood cells must be isotonic to one another. What do I mean? That the blood plasma and the red blood cells or white blood cells, maybe, they should have the same concentration of water and solutes in order for your blood to function normally, meaning they must be isotonic. In any case, there's variation in the concentration of water and solutes between the plasma of your blood and the red blood cells. Then we are going to have either hemolysis or crenation of the red blood cells, because osmosis is going to take place within your blood liquid, that water will either move from plasma into the blood cells, and then later on they will burst, or the water will move out of the blood cells which are into the plasma, and then they will crenate. And at the end of the day, your red blood cells cannot, they will lose their biconcave disc shape, they'll become crenated, or they'll become very, very, they'll burst, and then they cannot transport oxygen in your body. And the moment you cannot transport sufficient oxygen in your body, then you cannot respire. And then later on, the other thing is you will stop breathing, and then you will die. Therefore, in life, we ought to have to monitor the concentration of sugars, salts we take in our body, to monitor the blood sugar levels or blood salt levels, so that the plasma, the concentration of salts or sugars in your blood plasma is the same as that in the blood cells. Now, imagine they separated your red blood cells and they placed them in distilled water. What would happen? They've collected whole blood from you. They have separated the red blood cells. I'd request the producer to put the other picture on your screens. They get your red blood cells. They get whole blood from you, that one. And then after getting whole blood from you, they get the red blood cells, get a test tube containing purely distilled water. And then in this distilled water, they collect, they centrifuge your blood, collect the red blood cells only out, and then come and put them here in this test tube containing distilled water. What do you think will happen to you? what will happen after if you give this time? That is what is being displayed on your screens. You're having 3 test tubes. And in the first test tube, it was distilled water, but then later on they placed your red blood cells. And because the red blood cells are red in color, they are going to taint the color of the distilled water in that test tube to become pinkish, somehow pink. But because their red blood cells are being placed there. So after some time, the color of the water in the test tube, the distilled water in the test tube, will start turning to deep pink or deeper pink. Then that is the middle test tube. Then the third test tube, over time, the color of the test tube will turn red, the distilled water. Initially it was pink. It became deeper pink from the bottomwards. You can see it spreading up. And then later on, it becomes red, deeper red. What has happened? What has happened is what I am going to explain right now. It shows that when you get the red blood cells and you place them in distilled water, the concentration of water in the test tube is higher than the concentration of water in the red blood cells, meaning that the concentration, the distilled water, is hypotonic to the red blood cells. What makes the red blood cell have more solutes in them than the distilled water? Because inside the red blood cells, we're having the cytoplasm containing different organelles. We're having hemoglobin. We're having the, remember, mature red blood cells lack a nucleus. So there are components in the cytoplasm, a few components that make the concentration of the cytoplasm of the red blood cell more concentrated than distilled water. And as such, distilled water is hypotonic, and inside the red blood cell it is hypertonic. So what happens is the cell surface membrane surrounding the red blood cell will act as our semi-permeable membrane. So because there is more water into the test tube than the inside of your red blood cells that have been placed there, there is going to be osmosis take place, whereby water will move out from the test tube and it will enter into the red blood cell by osmosis across the plasma membrane of the red blood cell, which acts as our semi-permeable membrane. Now, as water enters into the red blood cells, as water enters into your red blood cell, now demonstrating it here more. As water enters into your red blood cell, it's going to keep on expanding or swelling or increasing in size to accommodate the incoming water. Remember, the plasma membrane of the red blood cell is our semi-permeable membrane. It is permeable to water. Water can easily pass through it. So as the red blood cell keeps on expanding to accommodate the incoming water, then it's going to reach a point whereby the cell membrane will be overstretched and it will reach its limit. That more incoming of water into the red blood cell will cause the plasma membrane, the cell membrane surrounding the red blood cell, to rupture or to burst. And when it bursts, the contents of the red blood cell will be spilled out. And of which of those contents is hemoglobin. And hemoglobin is what makes the red blood cell appear to be red in color. So the red hemoglobin pigment will be spilled out of the red blood cell into the water in the test tube. And that is done by all the red blood cell. That is why later on the color of the water of the test tube will change to red, showing that red blood cells have bursted. They've released their hemoglobin into the surrounding water environment, and then the entire water turns red. So that condition of red blood cells bursting to release their contents due to a lot of intake of water or influx of water into them by osmosis, that process is what we call hemolysis, or Hymolysis. It has different names, but we can take hymolysis. So that process is what you call hymolysis, and that is the effect of having a lot of water in your blood plasma compared to your blood cells. So that is what happens. This system is analogous to a balloon. You know, you get a balloon, you start blowing air into it. And remember, the surface of the balloon it's flexible, right? And it will reach a point when you blow air, it will keep on expanding to accommodate the incoming air. As kids always love that. So you blow, it will expand, you blow, it will expand. But the balloon reaches a point and reaches its limit. That further blowing of air into the balloon will make it to burst. And then you hear that sound, bah. When it bursts, then it means that the surface surrounding the balloon is weak, that it reached its limit, that it cannot extend anymore, that it will burst. The same that applies to your red blood cells. When they are placed in a dilute water environment, or when they're in an environment that has more water than them, water will enter into your red blood cells by osmosis. As water keeps on entering, they'll keep on expanding to accommodate the incoming water. And because the cell membrane is not rigid, it's weaker compared to the cell wall. When the more water enters the red blood cell, it will reach its limit. When it reaches its limit, it will burst, releasing its contents. And that is what you call hymolysis. Now imagine your blood plasma has less salts or has more water in it than your blood cells. What would happen? The same analogy idea would take place. But then this is when red blood cells are being placed in dilute water. But imagine the test tube where we are placing our red blood cell has a very high sugar concentration. Like we make a sugar solution. When I say we make a sugar solution, I mean we get water and we mix sugar, a lot of sugar in it, making it so concentrated or very sweet. Then when you get a sugar solution here, instead you can also make it salt solution, whatever it is. Sugar solution. Or it can be sugar or salt solution. Put it in your test tube. Then after, get whole blood, centrifuge it or spin it, separate the red blood cells, come and place them in our strong sugar solution. What do you think will happen later on? Will the sugar solution as well turn red, just like the 3 test tubes? Or it will remain colorless, or the color of the sugar solution, the initial sugar solution? So what will happen is when you place red blood cells in a very concentrated solution, this solution is called hypertonic. As I explained earlier on, the concentration of water in the surrounding sugar solution will be less than the concentration of water inside your red blood cell, meaning that the red blood cell is hypotonic to the surrounding water environment, or the red blood cell's cytoplasm is more dilute. It contains more water than the surrounding sugar solution. So what will happen? To magnify it more here, because the red blood cell has more water in it than the surrounding sugar solution, water is going to move out of the red blood cell by osmosis. And when water moves out of this red blood cell by osmosis, it's going to lose its shape. Because it's losing water, it will keep on shrinking, reducing in size. And then when it keeps on reducing in size, later on it's going to form a spiked shape, and it shows that it has crenated. Excessive loss of water will result into the red blood cell becoming crenated. Here, the red blood cell may not lose its hemoglobin to the external environment because it won't burst, but it will change its shape, it will get deformed. And when it gets deformed, space that accommodates hemoglobin has reduced. It cannot function normally. The hemoglobin cannot transport oxygen normally. And so the end result is crenation of the red blood cell, and also the red blood cell becomes deformed or dysfunctional, that it cannot carry oxygen. At the end of the day, you cannot carry out respiration because there is insufficient oxygen being carried by your body. And then in the long run, death will happen. So imagine those people who have high blood sugar levels, people who suffer from what you call diabetes mellitus, whereby they have issues with their insulin hormone from the pancreas, and then they cannot regulate their blood glucose levels very well. Every after meal, the concentration of glucose in their blood is very high, meaning that the concentration of the blood plasma is higher than the concentration of their red blood cells. They always have this condition. Their red blood cells always crenate. They lose their shape, and they reach a point where they cannot transport enough oxygen. Most of those people, when the diabetic and it goes to advanced stages, you look at them becoming very weak. They become very weak, that they do not have that much energy, because the red blood cells cannot transport enough oxygen that is going to be used in aerobic respiration to burn food to give them energy. So in the long run, they become weak. Now, what does that mean? That in our body, we ought to be so certain of the amount of water, amount of salts and sugars we take in our body, lest we have our blood cells either burst or hemolyze or undergo crenation, and thus we cannot transport sufficient oxygen in our bodies to support our body activities. So that is osmosis in red blood cells. Let us look at osmosis in plant cells. Now, plant cells, now in contrast to the red blood cells, the plant cells, in addition to them having a cell membrane, they also have a cell wall. Next, they have a definite shape than the red blood cells. So if you look at the shape of a plant cell, in most cases when you're drawing it, we give it shape. So it has some segment, it has the middle lamella somewhere. Then it has a central vacuole surrounded by a tonoplast. This central vacuole has very important osmoregulatory roles in the plant cell. So, and then it has a nucleus, which is in most cases at the edge. Then several other components like the mitochondria, the chloroplast, and whatever there. So, in brief, this plant cell has a very tough, rigid outer cell wall. It also has a cell membrane to add on that. In between the cell wall and the cell membrane, there is the middle lamella. So, when it comes to osmosis in plant cells, it's very hard for a plant cell to burst. Like in red blood cells, when it is placed in a dilute water environment. So imagine a plant cell is placed in a dilute water environment. Imagine it's placed in a hypotonic environment. Definitely, the last time I was here, I said, amongst freely permeable membranes that we have, that easily allow materials to enter and leave, one of them was a plant cell wall. The cell wall is freely permeable, the cell membrane is semi-permeable. So when you place a plant cell in an environment that has a lot of water, say distilled water, and maybe this is our beaker, then you placed it in distilled water. which is the same as our hypotonic or dilute water, what will happen is the surrounding environment will have more water potential than the inside of the plant cell. So therefore, the inside of the plant cell is hypertonic, the outside of the plant cell is hypotonic. If you understood the terms hypertonic, hypotonic, and isotonic, I repeated them. So when this is more dilute, is more concentrated, when the inner plant cell is more concentrated than the surrounding water environment, and then osmosis is going to take place. I'll request the producer to put that on the screen. Then osmosis is going to take place. Now we are looking at the last part where we're having turgid. What is going to happen? So here the plant cell is placed in a hypotonic or dilute solution, and then its contents are more concentrated than the surrounding solution. Osmosis is going to take place, whereby water will move from the outer environment inside into the plant cell by osmosis. As the water keeps on entering into the plant cell by osmosis, the vacuole, together with the cytoplasm of the cell forming the protoplasm, will keep on expanding due to more water entering it. If you can see on that illustration, the size of the vacuole in the first one, hypertonic, is very small, isotonic is medium, hypotonic is very big. So the vacuole will keep on expanding and it will keep on increasing in size to accommodate the incoming water. Then also the cytoplasm of the cell will start pushing. Because more water is entering, there will be a pressure that is going to push the walls of the plant cell to expand more to accommodate the incoming water. And that pressure is what I said we call Tagger pressure. Water enters, will cause the cell to keep distending to accommodate the incoming water. It reaches a point when there's a lot of water in here, and then the cytoplasm or the protoplasm will start pushing the cell wall further with a lot of force or pressure to distend them more as more water comes. As this pressure is what we call Tagger pressure. And because the wall of the plant cell, the cell wall, is very rigid, it's very strong, when it is distended beyond its limits that it cannot extend anymore, it will also exert an equal but opposite force. So the wall of the plant cell will also push inside. As it's being pushed to extend, it will also oppose by pushing inside, that I cannot extend anymore. So the force by which the cell wall of the plant pushes against the cytoplasm of the plant cell that is pushing it to distance, that is what we call wall pressure. So when the wall pressure is equal to the Tagger pressure, the plant cell cannot extend anymore, but it becomes so filled with water, so hard, so stiff, and turgid. And all the plant cells that are turgid will make the plant cell to appear to be firm, strong. That is why some small herbaceous plants, you find them standing erect on ground without any wood in them. But when you break it, you see water coming out of it. So what is making them to stand firm onto the ground is turgidity. So when Tagger pressure exceeds the wall pressure, the cell will keep on extending. Water will keep on coming. When Tagger pressure equates the wall pressure, more water enters, no water enters, no water leaves. So the plant cell becomes turgid, and that is what we call turgidity. But then what happens when it's placed in a hypertonic solution? The first drawing on your illustration. When it is placed in a beaker that contains maybe a sugar solution or a salt solution, then the opposite is going to happen, as you're seeing in the first drawing where we're having the word plasmolized. Instead of water entering, the outside hypertonic solution is more concentrated than the inner cytoplasm of the plant cell. So the plant cell has more water in it than the surrounding hypertonic solution. and as such, because the plant cell has more water in it, it's going to lose water by osmosis into the surrounding hypertonic solution. So the plant cell will lose water by osmosis. As it loses water, it keeps on reducing in size, shrinking itself, and then the wall of the protoplasm will pull away. As you're seeing, it's like star-shaped. The inner part, it's star-shaped. The wall of the protoplasm will pull away, and then it pulls away. The vacuole will also shrink, reduce in size, and then the plant cell becomes soft and flabby because it has lost a lot of water. And as such, such a plant cell is said to be plasmolysed. This can be done at home right now. If you have fresh Irish potatoes or pawpaws or mangoes, they're not the ripe mangoes, the raw mangoes. What you can do when you're eating a mango, actually, for us, those who live in villages, when you're eating a mango, why do you use salt or sugar to a raw mango? Because it's very sour, so you put there some salt. After you see water coming out. So that shows that water is coming out of the cells of the mango, or it's coming out of the cells of the mango because of osmosis. So when you peel a small Irish potato tissue, place it in as sugar crystals. Afterwards, you find water in the sugar crystals, meaning osmosis has taken place. So the importance of turgidity in plants is to support. It supports herbaceous plants to stand erect onto the ground, and that is very important in plant cells that do not have wood in them. That is what I wanted to tell you today. To reminisce about today, we've looked at osmosis in red blood cells, particularly the concept of hemolysis, the concept of crenation. Then we've looked at osmosis in plant cells, the concept of turgidity, and the concept of plasmolysis. And then we've said we should monitor the amount of sugar and salts we take into our body, lest our red blood cells hemolyze or crenate, and we cannot regulate our bodies so well. The reference question for today, as posted on your screen, is question number 31, UNEB 2015, paper 1. I used Macmillan Secondary Biology by Jeff Howard, 2000 edition. The next time I come here, I will not be teaching transport. It will be another topic of very interest, and I'll unveil it that very day. Thank you so much for your time, for God and my country.
Somera Muddiirolyo ng'ewagiddwa Maryland High School erisangibwa Entebbe. Ssomero ly'abawala n'abalenzi tusomesa arts and sciences okuviira ddala ku siniya esooka okutuukira ddala ku siniya ey'omukaaga nga lisangibwa mu kifo ekiweweevu obulungi. Tulina ebisuula eby'omulembe, science laboratory